Today we are going to talk about complement system. Complement. It consists of series of protein that complement the immune system and inflammatory reaction. So-called complement system. Complement protein is produced mainly by liver, but C1 is produced by GIT mucosa. Complement protein is present all the time in the blood, but it is present in inactive form. It is activated only when required. IgG and IgM can activate complement system by classical pathway. We can remember this as GM makes classic car. I will tell you later. Antibody can activate complement only when antibody is bound to the antigen. Antibody not bound to antigen, this cannot activate complement, but antibody bound to antigen, this can activate complement. Only IgG and IgM can activate complement by classical pathway. Now let us discuss complement protein 1. There are three component or three subunit of complement protein, which are Q, R and S. Out of these three, Q binds to antibody, that is IgG or IgM. Inactive complement protein is represented by C, but C bar indicates active complement. Now, in classical pathway, when C1 is activated, it activates C4, so that C4A component moves away and C4B component binds to C1A. Then C2B binds, then this C4B, C2B forms C3 convertase, which converts C3 into C3B. Now these all three together form C5 convertase, which converts C5 into C5B and C5A. Now this C5B leads to activation of C6, C7, C8, C9 respectively. Now these components C5, C6, C7, C8, C9 form membrane attack complex MAC, which form pore in the bacterial membrane. Intercellular contents leak and then bacteria dies. Activity of membrane attack complex is better in gram positive or gram negative. Activity is better in gram negative because gram positive bacteria have no outer lipid membrane, but gram negative bacteria have outer lipid membrane. So membrane is more accessible in case of gram negative. As membrane attack complex acts on the membrane, gram negative bacteria are more accessible for the damage by MAC. Now IgG can act as opsonin. Let us look here. C1, which activates C4B, C2B, C3B. Then C3B, this C3B also can act as opsonin, and IgG can act as opsonin. What is opsonin? Molecule that can bind one side to the bacteria and other side to the phagocytic molecule. One side to the phagocytic molecule, one side to the bacteria. Then this antibody is opsonin. IgG and C3B can act as opsonin. Classical pathway. In the classical pathway, IgG and IgM are involved. That is, GM makes classic car. We can remember this as GM. IgG and IgM makes classic car. That is classical pathway. This classical pathway is acquired immunity. But for alternate pathway, endotoxin by gram-negative bacteria. Endotoxins, that is by gram-negative bacteria, activate alternate pathway, which is example of innate immunity. Initially, there is activation of C3, so that C3B binds, and this activates B, so that BB binds and BA goes away. This activates C3B. Binds and C3A move out. Now this causes binding of C5B. These activate C6, C7, C8 and C9. This final component C5, C5B, C6, C7, C8 and C9 which from membrane latex complex is same in classical or alternate pathway, which is called final common pathway. When bacteria enters body for the first time, which pathway is activated? As when body faces bacteria for the first time, there is no formation of antibody, so that alternate pathway is activated. But once antibody is formed, classical pathway is activated. Classical pathway, GM makes classic car, C1, C4b, C2b, C3b, C5b, C6, C7, C8, C9. In alternate pathway, C3b, BB, C3b, C5b, C6, C7, C8, C9. Now there is another pathway called lectin pathway or MBL pathway. Surface of gram-positive bacteria have mannan protein. Our body has MBL, mannan binding lectin. In MBL, C4b binds, which activates C2. C2b binds, which activates C3. C3b binds, which activates C5. C5b binds, activates C6, C7, C8, and C9. This is the final common pathway. There is no C1, which is example of innate immunity. In classical pathway, there is C1. There is C1, C4b, C2b, C3, C5b, C6, C7, C8, and C9. But in lectin pathway, there is no C1, but there will be all component except C1 of classical pathway. C4b, C3b, C4b, C3b, C4b, C2b, C3b, C5b, C6, C7, C8, and C9. In alternate pathway, there is C3b, BB, C3b, C5b, C6, C7, C8, and C9. These C3A and C5A can activate the mast cell and help in anaphylaxis, so amplify the inflammatory reaction. C5A can act as chemotactic agent. Membrane attack complex formed by C5b, C6, C7, C8, and C9, which causes membrane lysis, and its activity is more in gram-negative bacteria because membrane there is outer membrane present in gram-negative bacteria, so that membrane is more accessible to MAC. C3b acts as antibody producing amplifier. It activates B cell. GM makes classical CAR in classical pathway. Classical